This training session will cover sample handling and coordination with the lab. The topics we will be covering include personal protective equipment, sample handling and storage, sample containers, sample preservation, sample hold times, sample labels, the chain of custody document, EPA Method 5035, getting your samples to the lab, quality control, and pre-field preparation. Let's start with personal protective equipment. Your PPE is intended to protect both you and your samples. Gloves protect you from exposure to the chemicals that may be in the samples and from exposure to sample preservatives. Gloves also protect your samples from being cross-contaminated by chemicals on your hands. You should change your gloves often. It's generally considered best for you to wear 2 layers of latex gloves and to change them between samples. Wearing eye protection helps prevent soil, water, and preservatives from harming your eyes. It only takes a second for contaminants to be splashed or otherwise enter your eyes if you're not wearing eye protection. Next, we are going to discuss sample handling and storage. As with all aspects of field work, safety should be your first concern. You also need to think ahead of time to make sure that you do not bias your sample results. Sample bias can result from the way you collect your samples or even where you set up your work area. When you're collecting samples, you generally want to get samples that are representative of a larger area. This is why you sample from multiple locations within a soil stockpile or from multiple drums and then composite those samples. When you decide where to set up your sampling station, you need to think about your proximity to things that could bias or cross-contaminate your samples. For instance, you don't want to be handling your samples near the exhaust from equipment. You also don't want to set up downwind of parked cars, active dispenser islands, or the drill rig. Other things that can contaminate your sample include things like the sunblock you are using, hand sanitizer, or tape used on sample containers. Sunscreen often contains a high percentage of alcohol. For example, this brand has up to 65% alcohol by weight. Likewise, hand sanitizer can often contain upwards of 60% ethyl alcohol. When we are looking for part per billion range TBA, also an alcohol, you can see how this could easily contaminate your samples. You also need to be aware that your samples can be cross-contaminated by not sufficiently cleaning your sampling equipment between samples. You should also keep samples from coming into contact with each other or the ice melt water in your cooler. This is why samples should be stored in Ziploc bags. Now let's take a look at sample containers. We use a wide variety of sample containers. The type of sample container is based on the media you will be collecting and what analyses you require. You may be using brass or stainless steel tubes, Encore or TerraCore samplers, glass bottles, plastic bottles, Tedlar bags, or Suma canisters. If you have any doubt about what type of containers are required for your project, talk to your client manager, the PG for the project, or the lab project manager. Keep your sample containers clean and cool. Don't store them in areas where they could get contaminated prior to you even collecting your samples. Don't use old containers if you can help it. Order the containers you need for your job and get new ones for the next job. Know which container is needed for which analysis. Know what minimum sample volume is required for your analysis. Generally speaking, your container should be completely filled. If you are sampling for volatiles, you do not want headspace in your sample, whether it's for water sample in a VOA or soil sample in a tube. Now let's talk about sample preservation. Preservatives are used to slow the degradation of your sample and increase the time allowed for holding and analyzing the samples. The bottles you get from the lab will usually come with a pre-measured amount of preservative in them. The type of preservative will be specific to the analysis method. Because these bottles contain preservative, you obviously do not want to rinse out the bottles before you use them. You want to completely fill the bottles, but you do not want them to overflow. Make sure you use plenty of ice in your cooler. This is obviously very important with VOC samples, but other analyses are also temperature critical. You want to make sure that you are wearing your proper PPE, because the preservatives are often acid or other chemicals that are not friendly to your skin or eyes. Now we're going to discuss sample hold times. Your hold time will vary depending on the analysis requested, whether your samples are preserved or not, and the type of container. Typical hold times can vary from as long as 180 days for some metals to as little as 15 minutes. For volatiles, water samples generally have a hold time of 14 days if they are preserved, or 7 days if unpreserved. Soils collected in Encore samplers have a hold time of 48 hours. If the samples are frozen, the hold time can be extended up to 14 days. Don't confuse hold time and turnaround time. For example, vapor samples typically have a maximum hold time of 72 hours. This means the lab has 72 hours from the time you collect the sample until they must analyze the sample. However, if you don't need the lab report immediately, you would not request a 72-hour turnaround time. Typically, you don't need your results until a standard turnaround of around 2 weeks. Requesting an unnecessarily short turnaround time significantly increases the cost of your lab analysis. If you're going to be collecting samples with a short hold time, you should notify the lab in advance so that they can be prepared to handle those samples. Obviously, you should try not to collect short hold time samples on a Friday or before a holiday. Sample labels. Your samples are no good to anyone if you don't know which one is which. Your labels must be legible. If the lab can't read the label, you won't get the proper analysis. Make sure you use a pen that will not run. We generally use fine-tipped Sharpies. Make sure that the sample IDs you use follow the naming protocols required by Wayne Perry and GeoTracker. If you have any doubt, ask your client manager or the PG. The chain of custody document. Chains of custody are legal documents and must be completed correctly. Your entire sampling for a site can be rendered invalid if the COC is not properly completed. Like sample labels, the COC must be legible and include all required information. Your chain of custody should be filled out at the time of sampling. The chain of custody documents the physical custody of the samples from the time of collection through analysis. If you wait to fill out the COC until the end of the day, or when you get back to the office, you will have jeopardized the integrity of the custody chain. Never use whiteout. If you make a mistake, you should simply draw a line through the mistake, enter the correct information, and initial the change. When you have a long list of analyses, it's tempting to attach another sheet describing the analyses and simply put "See attached" on the chain of custody. This can have legal implications because it is technically not part of the official signed chain. It also can get lost in the process. EPA Method 5035 is a method of collecting and preserving soil samples for volatile analyses. It is typically required for any site in Los Angeles or Ventura counties, and for any DTSC or RCRA site. The most common way to collect samples for 5035 involves the use of the Encore sampler. Encores are small plastic samplers used with a metal T-handle. You typically have to collect from 3 to 5 Encores per sample. Because of this, the cost of the Encores can get quite high, since they typically run from $7 to $10 per Encore. The T-handle runs about $75. As we discussed earlier, you need to be aware of the hold times associated with Encores, especially if collecting samples on a Friday or before a holiday. Another option is through the use of the TerraCore system. The main difference between the TerraCore and Encore systems is that the TerraCore requires your samples to be preserved in the field using methanol or sodium bisulfate. With either system, you should never reuse the samplers. Once you have collected your samples, you still need to get them to the lab. Make sure that you use the right size cooler. Don't have a 40-gallon cooler with 2 sample bottles rattling around inside. Likewise, don't cram so many samples into a small cooler that you don't have room for a sufficient amount of ice. It's best if you can bag your ice to keep the ice melt water from coming into contact with your samples. At a minimum, your samples need to be bagged. This will protect your samples and the information contained on the sample labels. Don't use blue ice. You won't get sufficient cooling of your samples. Don't put glass bottles in contact with each other. If you have plastic bottles too, put a plastic bottle between your glass bottles so you don't have glass-on-glass contact. Take advantage of the free lab couriers. If you call them in advance, you can have them come directly to your job site to pick up samples. This can be a big help if you have a long sampling job. Know what the sample receiving hours are for the lab. It's your responsibility to make sure that your samples do not exceed the hold time, and if you miss the drop-off times, you could find yourself in trouble. Sample quality control. Sample quality control starts at the time of sampling and continues through the lab work. You need to know if your project requires trip blanks, equipment blanks, field blanks, or temperature blanks. If you are unsure, ask your client manager. The blanks will be provided by the laboratory with your bottle order if you ask them. You also need to know what types of duplicates may be required. The lab is required to have a matrix spike duplicate as part of their QA/QC procedures. These MS-MSD samples can be made up in the lab, but you will get better site-specific results if you can provide the lab with a field duplicate MS-MSD sample. For more information on this, talk to your client manager or your lab representative. Pre-field preparation. Proper pre-field preparation will make your sampling event go much more smoothly. Know why you are collecting the samples. Are you doing it to delineate the site or for evaluation of remediation progress, or to support closure? If you know how the data will be used, you can do a better job of collecting the samples. This is why you should try to write all of your own reports. By having to analyze the data and write the report, you quickly become more familiar with how sample collection can influence the results. You also need to know what the expectations of the client and agency are. By knowing what is expected, you will be better able to make decisions about whether step-out borings are needed or urgent notifications are required. Know the reporting limits that are needed. It will do you no good to do all the work and have your results all be ND less than 5,000 when the action level is 13 ppb. If you talk to the lab in advance, they can often adjust their procedures to get you the reporting limits you need. Know what deliverables are required. Do you need an electronic deliverable for GeoTracker, Wayne Perry, or the client? Again, this needs to be discussed in advance. In conclusion, if you have a better understanding of the proper sample collection and handling procedures, and if you coordinate your efforts with the laboratory, your sampling efforts will go much more smoothly and keep everyone happier. If after watching this video you still have questions concerning sample handling and lab coordination, please feel free to discuss your questions with your client manager, the PG for your projects, or your lab representative. Thank you.